A very good morning. Welcome in the class. The topic of today's class is isolation of DNA and RNA. So as we say in the starting, let's crack CSIR UGC NET together. You're watching this video under the platform of Unacademy. Unacademy is one of the India's largest platform for the preparation of CSIR UGC NET Examination. I'm your educator Farida Johar Bandukwala. I have teaching experience of 10 years and I've qualified MPEC examination in year 2018. If you're opting for Unacademy, what all things you are going to get? You're going to get daily life classes so that you can always be in that momentum of studying, and live classes you can ask your doubts directly with the faculty members. The whole syllabus is quite structured so that you can complete all the 13 units in a small span of time, and live tests and quizzes are taken so that you can evaluate yourself that how much you have learned so far. And whichever category you are opting for, then you will get the unlimited access towards all the videos and all the study material which has been recorded in that particular category. Unacademy gives free live classes at two platforms. One of it is YouTube. You can watch the free live classes on YouTube directly by subscribing the channel, that is Let's Crack CSIR UGC NET, and you can watch the special class that is also free class by downloading the Unacademy learning app. After downloading the app, make your account over there, and then you will be asked to fill the referral code of an educator. So fill my referral code, that is FB01. By filling my referral code FB01, you will be getting the access to watch all the free special classes of mine and of other educators. After that, there is this plus subscription. This plus subscription is a paid subscription. You need to pay a certain amount for this. And this plus subscription, recently a discount is going on. So for 12 months, you need to pay 11,550 rupees, and for 24 months, you need to pay 16,800 rupees. And still, the things are not over. You can get an additional 10% discount if you're using my referral code FB01. So use my referral code FB01, and by using my referral code, the 12 monthly course is going to cost you 10,395 rupees, and for 24 months, it is going to cost you 15,120 rupees. So this was the introduction about Unacademy. Let us begin with the topic of today, that is isolation of DNA and isolation of RNA. So first of all, we'll be talking about the isolation of DNA. So DNA isolation is a process of purifying the DNA from the sample using combination of physical and chemical method. So isolation of DNA is nothing but purification of DNA. Purification of DNA by physical and chemical methods. After that, the first isolation of DNA was actually done in year 1869 by Michener. So the first isolation of DNA was done by Frederick Michener. First DNA was isolated in 1869 by Frederick Michener. So the methods used to isolate DNA, it depends on the source of DNA, then age of the sample, then size of the sample. So method of isolation depends on source, age, and size of sample. So isolation of DNA is needed for what all things that we need to know. So it is needed for genetic analysis, which is used for various scientific, medical, and forensic purposes. So isolation of DNA is needed for genetic analysis. in the field of scientific, medical, and forensic purpose. Right? So scientists, they use DNA in various applications other than the scientific, medical, and forensic purposes. They use to introduce the DNA into cells and animals or plants for various diagnostic purposes. And in medicine, later on, the application of such introduction is also done. So on the other hand, the forensic purposes and forensic science needs to recover the DNA sample from an individual or from the site of accident, in case of robbery, in case of murder. So in forensic field, all those things can be solved by isolation of DNA. Then presence of various proteins, lipids, polysaccharides, and other organic and inorganic compounds in DNA interfere in isolating the DNA, because DNA is not kept somewhere safely that yes, this is DNA, go and pick it up. It is present in association with so many other things, like proteins are there, lipids are there, polysaccharides are there, those all things are there. So those all things need to be removed. So it's not that easy job. They can also then it can also in these all things, that is protein and all these all actually interfere in the quality of DNA and the amount of DNA which is obtained from the source. So all these things should be kept in mind while isolation. Now talking about the source, that what all things can be the sources of DNA isolation. So source. So the source can be diverse. Every living or dead cells can be a source of DNA. Every living or dead cell can be source of DNA. Then common source for DNA isolation would include the whole blood cell, then hair cells, then sperm, bones, nails, tissues, blood stains, saliva, buccal cheek cells, then epithelial cells, then urine, then the paper cards which is used for the sample collection, then bacteria, animal tissue or plant. These all can be the sources. So we would just write some of them. Whole blood, hair, follicle specifically, then sperm, bones, etc. can be source of DNA. Then stored samples can come from archived tissues such as frozen blood or tissues, ex-human bones or tissue, ancient humans, animals or plant samples. So stored samples can also serve as source of DNA. Now after source, procedure. So isolation of DNA, it basically consists of four major steps. So first is preparation of a cell extract, preparation of a cell extract, then purification of DNA from cell extract, then concentration of DNA samples. We'll be elaborating each and every step, so do not worry at all. These are the main steps we'll be elaborating. Then measurement of purity of DNA concentration. Measurement of purity of DNA concentration. So we are going to elaborate each of this step. The first step is preparation of a cell extract. The preparation of a cell extract. Now, to extract DNA from a tissue or a cell of interest, the cell have to be separated and the cell membranes have to be disrupted. So, first of all, we'll be writing cells to be separated and cell membrane disrupted. After that, extraction buffer, it helps in carrying out these processes. So extraction buffer will help in separation and disruption of cell membrane. After that, chemicals such as EDTA are needed. So EDTA, what is its full form? EDTA, it is ethylene diamine tetraacetate. Ethylene diamine tetraacetate. So this thing is needed because it removes the magnesium ions, which are actually essential for preserving the structure of cell membrane. So EDTA removes magnesium ion, which maintains structure of cell membrane. Then another thing is needed, that is SDS. The full form of SDS is sodium dodecyl sulfate. So the sodium dodecyl sulfate helps in disrupting the cell membrane by removing specifically lipids from the cell membrane, and thus it is included in extraction buffer. So SDS removes lipids from cell membrane. So these both of the things are part of extraction buffer. Now, when the cells are lysed, the final step is the preparation of cell extract. That is the removal of insoluble cell debris. So after lysed cells, removal of insoluble cell debris is done. So cell debris and other partially digested organelles can be pelleted by centrifugation, leaving the cell extract as a reasonably clear supernatant. So the whole thing, the cell extract will be actually centrifuged and the pellet would be containing the cell debris and partially digested organelles, which will be discarded, and the clear supernatant would be kept. So next is centrifugation of cell extract, pellet discarded and supernatant kept. This will be done in the preparation of a cell extract. After preparation of cell extract, the next step is purification of DNA from cell extract. Purification of DNA from cell extract. Now, how is it purified? So, in addition to the DNA, the cell extract is not going to contain just DNA. It is going to contain certain other things also. It is going to contain significant amount of detergent, protein, salts, reagents used during the lysis step, and it will contain specifically RNA. So a variety of procedures would be used to remove the contaminants and leaving the DNA in pure form. So we'll be just noting down these two things, and then we'll be moving towards the procedure which is used for removal of all these things. So in addition to cell extract, in addition to DNA, or let us write along with DNA, cell extract also contains detergents, proteins, salts, reagents, and RNA. So various steps would be taken to remove them. So the most commonly used procedures for these are most commonly used procedures are ethanol precipitation, ethanol precipitation, then phenol chloroform extraction, then MiniColumn purification. Right, so we'll be elaborating these steps: ethanol precipitation, phenol chloroform extraction, and MiniColumn purification. So first is ethanol precipitation. So ethanol precipitation step is done by using ice cold ethanol or isopropanol. So in ethanol precipitation, ice cold ethanol or isopropanol is needed. So why is it needed? Because DNA is insoluble in these alcohols. So it is going to aggregate together and giving rise to a pellet upon centrifugation. That is why it is used. So DNA is insoluble in these alcohols. Thus, it will aggregate and form pellet upon centrifugation. Then precipitation of DNA can be improved by increasing the ionic strength. Usually addition of sodium acetate is done so that the DNA precipitation can be improved. DNA precipitation is improved by adding sodium acetate. So in ethanol precipitation, what words you need to remember is use of ethanol and isopropanol. And why so? Because DNA is insoluble in them. So they are going to form aggregate, and the whole process can be improved by addition of sodium acetate. So this was ethanol precipitation. After this, we need to see phenolchloroform extraction. Phenolchloroform extraction. So phenolchloroform extraction is that kind of extraction in which the phenol is used for denaturing the protein in the sample. So phenol denatures protein, and after centrifugation of sample, the denatured proteins stay in the organic phase while the aqueous phase contains the nucleic acid, and it is mixed with the chloroform, then which removes the phenol residues from the solution. So phenol is used for denaturing the protein, and after centrifugation, denatured proteins they stay in organic phase. Denatured proteins they are obtained in the organic phase, and the aqueous phase and the aqueous phase it is going to contain the nucleic acid. Right. So this aqueous phase will be mixed with chloroform, so that the phenol residues can be removed. So that phenol residues are removed. So denatured protein and nucleic acid. Now that last thing that we need to see in this category is mini-column purification. Mini-column purification. Now, mini-column purification is a purification which actually relies on or depends on the fact that the nucleic acid may bind to the solid phase. Absorption of nucleic acid may occur on the solid phase such as silica or some other solid phase depending on the pH and the salt concentration of the buffer. So that is how it can be purified. So binding of nucleic acid, that is adsorption to solid phase such as silica, and will depend on pH and salt concentration of buffer. So this was mini-column purification. Then we will be talking about concentration of DNA samples. So the third step is concentration of DNA samples. So the most commonly used method for concentrating the DNA sample is use of ethanol, and it is called as ethanol precipitation. Done by ethanol precipitation. So in the presence of salt and at temperature of minus 20 degrees Celsius or maybe lesser than that, the absolute ethanol will efficiently precipitate the polymeric nucleic acids. So in presence of salt and temperature minus 20 degrees Celsius or lesser than that, absolute ethanol will precipitate the DNA or nucleic acid. So with a with a concentrated solution of DNA, one can use a glass rod to pull out the adhering DNA strands, while to dilute solution, precipitated DNA can be collected by centrifugation and it can be redissolved in an appropriate volume of water. So we'll just write over here concentrated solution of DNA. Glass rod is used to spool out DNA and in dilute solution of DNA, centrifugation is done. Centrifugation is done and then the DNA is collected. So this is how the concentration of DNA sample is done. And lastly, we need to know that what is the concentration of DNA sample that has been obtained. So it is called as measurement of purity of DNA concentration. So fourth is measurement of purity of DNA concentration. So DNA concentration can be accurately measured by UV absorption spectrometry, measured by absorbance spectrometry. The amount of UV radiation which is absorbed by a solution of DNA is actually directly proportional to the amount of DNA sample. So amount of UV radiation absorbed is directly proportional to the amount of DNA. So usually the absorbance is measured at 260 nanometer. Measured at 260 nanometer. So at this wavelength, the absorbance of absorbance of 1 corresponds to 50 microgram of double stranded DNA per ml. Right, so it is measured at 260 nanometer. Remember this is much more important. And absorbance of 1 is equivalent to 50 microgram of double stranded DNA per ml. So this is like the thing which is said. So with a pure sample of DNA, the ratio of absorbance at 260 nanometer and 280 nanometer is 1:8. So this is also an important thing. So ratio of less than 1:8 indicate that the precipitation is contaminated either with protein or with phenol. So let us write with pure sample of DNA, the ratio of the absorbance at 260 nanometer and 280 nanometer, that is called as A260 to A280, is 1:8. Now ratio less than 1:8 is going to indicate that the precipitation is contaminated. Ratio of less than 1:8 indicate that precipitation is contaminated. Now this contamination can be due to protein or phenol. Though chloroform was used to remove the residues of phenol, but still if it is present, it will give a ratio of less than 1:8 when it is absorbed at 260 and 280 nanometers. So we have done DNA isolation. Let us just have a quick revision of DNA isolation. So the purification or the isolation of DNA, it is done by physical and chemical method. Then the method of isolation depends on from what is the source, what is the age, and what is the size of sample. Then why it is needed? It is needed for scientific, medical, and forensic purposes. Then source can be anything, living or dead cells, stored samples, hair follicles, sperm, bone, nail, uh, saliva, urine, anything can be the sample. Now the procedure consists of 4 steps. First is preparation of a cell extract, then purification of DNA from cell extract, then concentration of DNA sample should be measured, then measurement of purity of DNA concentration is done. So first is preparation of cell extract. So cells are separated and the cell membrane is disrupted, and extraction buffer is used for the above process. So EDT is responsible for removal of the magnesium ion and SDS is needed for the removal of lipids. So when the cells are lysed, the insoluble cell debris are removed, and centrifugation is going to give us the supernatant, which contains the nucleic acid, and rest of the things will be removed out. After that, in the second step, that is the purification of DNA from cell extract. Along with DNA, the cell extract would contain lots of detergents, proteins, salts, and reagents. So all these things need to be removed. So 3 steps are taken for this. First is ethanol preparation, then phenolchloroform extraction, then MiniColumn purification. So in ethanol precipitation, ethanol or isopropanol is used in a very chilled form, and DNA becomes insoluble in them. And if sodium acetate is added, it improves the whole process of precipitation. Then phenolchloroform extraction. Phenol is used for denaturation of the protein. Phenol is used for denaturating the protein, and centrifugation is done. So denatured proteins will be obtained in the organic phase, and the nucleic acid would be obtained in the aqueous phase, and chloroform is used for removal of the phenol residues. So this is phenolchloroform extraction. Then MiniColumn purification is done, that is binding of nucleic acid to the solid phase is done, and the binding will depend on pH and salt concentration of buffer. Then concentration of DNA sample is done by again ethanol precipitation. So in the presence of salt and at a very low temperature, -20°C, the absolute ethanol will actually precipitate out the DNA. So concentration of DNA. So when the DNA is very much concentrated, it can be actually wound on the glass rod, and if the solution is dilute, it can be centrifuged. Then measurement of purity of DNA is done at 260 nanometer, and then ratio of absorbance is done, that is at 260 and 280 nanometer. The ratio should be 1:8, but if the ratio is less, it means the whole sample is quite contaminated. Right. Now coming to the second part of the class, that is the isolation of RNA. isolation of RNA. Right. So we have seen that how the DNA is isolated. The isolation of RNA is a little different than DNA. Why is it different from DNA? Because the RNA is not as biochemically stable as DNA. So we'll write purification. Purification is different than DNA because RNA is not very stable. Right. So because RNA is also in single stranded form, any hydrolysis event can break the phosphate backbone, which is going to result into cleavage of molecule into fragments. So its single stranded nature makes it vulnerable to breakage. So two factors which contributes to the biochemical instability of RNA. So first is the RNAs that are very stable enzymes and cannot be easily inactivated. So instability of RNA goes to first is presence of RNAs, presence of sturdy RNAs, sturdy RNAs. So sturdy RNAs are actually very much stable, so they cannot be easily inactivated, can't be inactivated. Then second is RNA is thermodynamically less stable than DNA. Thermodynamically less stable. So less stable as compared to less stable than DNA. Why? Why is it less stable than DNA? Because of the 2' hydroxyl group on the ribose ring, which promotes the hydrophilic attack of 5'3' phosphodiester bond from the 2'3' to form the 2'3' cyclic phosphate. Thus, it is thermodynamically less stable. So, we need to justify this point. Okay, we'll just use this page only. We'll use another ink. So, it is thermodynamically less stable because of 2' hydroxyl on ribose. So this 2' hydroxyl on ribose is going to promote the hydrophilic attack of the 5'3' phosphodiester bond. So it promotes hydrophilic attack. It promotes hydrophilic attack on the 5'3' phosphodiester bond to form 2'3' cyclic phosphate. Therefore, if RNAs, even if RNAs are eliminated, if we get rid of RNAs, then too this RNA purification can be inhibited because of the RNA can spontaneously degenerate itself in the solution. So no matter if you are getting rid of RNAs, then too the problem can arise. Now, if the problem is arising but we want to isolate DNA, we need to look for some other thing. So to overcome this process of its self-degeneration, this biological decay of RNA, purified samples are stored at minus 20 degrees Celsius as ethanol precipitates. So storing it at a very low temperature can solve the problem to an extent. So to overcome biological decay of RNA, purified samples are stored at 20 degrees Celsius as ethanol precipitates. Okay, are you understanding? I think we are together till here. Now, the purification of mRNA, it involves two basic steps. The two basic steps we are going to see. So, two basic steps of purification. So first, biochemical separation of total cellular RNA from DNA and protein using a strong protein denaturant to inhibit the cellular RNAs to just get rid of DNA and protein. So first thing, biochemical separation of total biochemical separation of total cellular RNA from DNA and protein using a strong protein denaturant to inhibit the cellular RNAs. By using strong protein denaturant to inhibit cellular RNAs. First thing. Now second step, isolation of poly-A tail mRNA using an oligo-DT affinity matrix. So a common method used to isolate mRNA from the tissue culture we'll be seeing. So it is done by guanidinium thiocyanate, and guanidinium thiocyanate is a strong protein denaturant that lyses the cell and inhibits the cellular RNA. So we'll be seeing a flow diagram, but before that, the two basic steps. First is biological separation of total cellular RNA, and second is to use of poly A tail of RNA to separate it on oligo DT affinity matrix. These are the two important things that we have seen. Now, a common method which is used to isolate the mRNA from tissue culture that we are going to see now. And the method uses uses strong denaturant, that is guanidinium thiocyanate. Guanidinium thiocyanate, and it actually inhibits cellular RNAs. This is the thing. Now we'll be seeing the flow diagram of this method. So we have got a cell culture. To this cell culture, we'll be adding guanidium thiocyanate. We'll be writing the abbreviation. Guanidium thiocyanate is added. After this, there will be addition of phenol and chloroform. After this, the whole thing would be centrifuged. Then we'll be getting aqueous phase and the another phase. So removal of aqueous phase will be done. Removal of aqueous phase. Now the whole work will be done on aqueous phase. So aqueous phase will be obtained. So we'll just continue there from there. So aqueous phase. In this aqueous phase, there will be addition of isopropanol. Then it will be again centrifuged. After centrifugation, we'll be getting the RNA pellet. When we'll be getting the RNA pellet, it will be separated on the oligo DT column. Separation of mRNA with oligo DT affinity matrix is done. So this is the method. So isolation and purification of mRNA from tissue cell culture using the guanidium thiocyanate and oligo DT matrix or oligo DT cellulose column is done. This purification method is actually based on finding the RNA preferentially partitions to the aqueous phase in the solution containing the guanidium thiocyanate, and this is done at pH 4 pH 4 in the presence of phenol and chloroform. pH is 4 in the presence of phenol and chloroform. So under these conditions, the protein partition to the organic phase and most of the large DNA, it is trapped in the interphase. So remember that the protein partitions into the organic phase and DNA is trapped in the interphase. So poly-A mRNA is purified from the total RNA by using the polyadenylate polyadenylated affinity ligand, that is the poly-D ligand. So this is how the whole process of RNA purification or RNA extraction is done. So in isolation of RNA, what all things we have seen, that it is different than DNA isolation because it is not very much stable. Why? Because of its single stranded nature and presence of sturdy RNAs and its thermodynamically instability, that is it itself gets denatured because of the presence of 2-OH. The 5-3- phosphodiester bond is attacked to form 2-3- cyclic phosphate. And to overcome this, the whole sample is stored at -20 degrees Celsius as ethanol precipitates. So there are two things which are used in this basic purification of RNA. So first is biochemical separation, in which DNA and protein should be separated out, and in second thing, the whole RNA extract should be used for the separation of just mRNA. So second, you can say DT affinity matrix. So method, we have seen that the cell culture would be taken, in that guanidium thiocyanate would be added and phenol chloroform would be added. It will be centrifuged. Removal of our RNA would be done in the aqueous phase. Protein will be gone out in the organic phase. DNA would be trapped in the interphase. So aqueous phase will be used. In that, isopropanol would be added. It would be centrifuged and RNA pellet would be obtained. Then it will be again solubilized in the buffer, and then separation of this mRNA, because of its poly tail, it will be separated on the DT affinity matrix. So this is how the whole process of RNA purification occurs. And like this, we have completed the topic of isolation of DNA and isolation of RNA in a systematic manner. And as we say in the starting and end, let's crack CSIR UGC NET together and hit the bell icon for getting all the notifications of the upcoming videos. And if you haven't subscribed our channel yet, subscribe it. And if you really like my video, do not forget to give it a thumbs up. It really motivates the educators like us. And use my referral code FB01 for getting the plus subscription. Recently a discount is going on, so if you're planning for qualifying the examination, go for the plus subscription and use my referral code for getting a 10% discount. And in a special class also, you can use my referral code to attend the special free classes. So till we meet next time, that is 5 p.m. in the evening. Thank you very much for your patient listening. I want to see you all at 5 p.m. again, and tomorrow 10:30 with some another interesting topic of CSIR, and evening we are doing the topics of GATE syllabus. Hope to see you all at 5 p.m. today and tomorrow 10:30 for an interesting topic of CSIR, and in the evening interesting topic of GATE. So this is done for now. We'll be meeting. Till then, be happy and keep on studying.